This still shows up on mine. Regardless, yeah, I would just throw it at. It's perfect. Well, this is part of the.
Yeah, I think it's the phone box though. That's exactly what happened yesterday. Said he would come in kind of a budget guts for the street. Yeah, we're starting to short line. Well, Dan Garrett is my level. Oh yeah, that's right. Yeah. He also was chair during that time. Dan Garrigan was there. Is that right? Yeah, he became chair. And then he was, why would you work in a cafe? He lives right here. Maybe to get away from people. Maybe he just didn't like that guy. He's in right now in. He went to Rochester. Yeah, no, but he's. Yeah, he's in. He's in conference with. He hasn't quite quit. A bit of research. He's doing a lot of research. It's pretty good. Segregation distortion using data. He's got time. Yeah. Research. And his wife Sarah also works for a company. Where was he when you? We found the first hint even about we got vindication. Yeah, I know what we didn't call it. But another thing that is interesting. I've started, it's really unlikely that when you look at it, we'll just wait a couple more minutes. There's still a number of people in line, so. Yeah, there's 20 people in line. Funny how it happens. Took an engineer to figure that out.
You never know why places always set it up.
Yeah. Educated.
But again, you can do all that. I'll do all that. I will work that out because I'm going to talk about that. Okay? So this and that. Okay. Okay?
Graphic models, model substitution.
Okay, I think we're going to get started. I think everyone's almost in, and you know, we're so excited. This is our inaugural event. Rasmus, thank for opening us up here. It's just wonderful to see all of you out here at 5:30. We're going to have a wonderful evening. Rasmus is going to talk to us. Michael will introduce him in a second. Going to have a wonderful evening. His lecture, and then some wine and cheese, yay! And then we'll follow that up with a local panel discussion, and Michael will talk a little bit about that in a second. I want to acknowledge a few folks. Again, this is the first of 7 of these taking place roughly each month, with the exception of the summer. So welcome to the first one. You know, I want to acknowledge our Bio5 director and our leader, Jennifer Barton, for allowing us to do this and making this possible. I also want to acknowledge Michael Hammer and the rest of the associate directors. We've been planning this for quite a while, and we know this is going to be a very important event for the university and the med school. So, as I said, we're really excited. The Bio5 staff, I won't get them all, but I know Kelly and Amy and Tatiana worked really, really hard at the last minute. There were a lot of events going on at Bio5, and so they really worked hard to put everything together that you see tonight. So would everybody give them a hand, first of all? So I'd like to start out, and I'll spend 3 or 4 minutes just kind of giving you the rationale for why we have this symposia. Why do we have the Precision Wellness Initiative? What are we trying to do? And in one word, we're trying to operationalize this very, very complex area of precision health and precision wellness. If you go anywhere and everyone says they do precision health, and you go, okay, what do you do? What is precision health? And we're trying to kind of codify that and operationalize it so it makes sense. One of the reasons it's so extraordinarily complex is it involves so many disciplines. So this is our model, our transdisciplinary model, to create conceptual and translational innovations in precision wellness. And we really start at the top, and that's why we have Dr. Nielsen here tonight. We start at the top, early in environments, nutrition, climate, altitude, and the diversity of those environments, and how humans evolved and adapted to those environments. And so we believe that if we understand our beginnings, then we'll be able to place our present into context. And so that's why it's really important to have these disciplines in human adaptation involved here. Again, they gave rise to genetic, epigenetic, and microbiome variation within and among human populations. Then within the last 75 years, we saw the emergence of the modern Western diet. We saw contemporary nutrition, contemporary lifestyles, and we see populations that represent all ancestry. They clash with this variation. They create gene-diet interactions. These gene-diet interactions, with that we can understand humans now, and particularly the gene-diet interactions that drive human disease. From that, we go through transcriptomics, proteomics, and down to metabolomics, and we're really making a concerted effort here to understand metabolomics and lipidomics because we believe that those will be the intermediate molecular phenotypes that take us to the promised land, that take us to the human phenotypes. So we think it's critical to understand the biochemical networks that drive disease and the biomarkers that indicate disease status. So we have a real focus here as well. None of this can be put into any context unless we know how to deal with all this big data. And one of the things, I've been here 6 months from Wake Forest, and one of the things that attracted me here, aside from Bio5, but also within Bio5, is all the data science here. And the data science capabilities to put all of this together and put it into a conceptual framework that makes sense is really an incredible strength here. So to be able to understand all and make sense of all of this complex data, and again, the computational approaches that integrate the biological systems. Finally, as someone at a talk said to me not long ago, all that fancy stuff don't make a bit of difference if you can't get people to change. So we have to be able to understand behavioral models of change, and we that has to be precision as well. Different cultural backgrounds, different ancestry backgrounds, different things for different folks. So that's the final step to implementing precision wellness. And so we have to understand how to change human behavior to take advantage of all the progress. So that's our multidisciplinary model to create conceptual and translational innovations in this area. We believe they're all necessary. We believe we all have to come together to be able to understand something this extraordinarily complex. And we're just so thankful that Bio5 serves as Switzerland to bring us all together where there's no boundaries and we can all come together and work in this translational model type way. With that, I'll hand the microphone to Michael Hammer. Michael?
Thanks, Guy. Today's speaker, RasmusNielsen, we're very lucky to have him here. He's really a leader in so many areas to do with the middle box here, the variation within and among human populations. We're all different, so starting with the precision wellness model, it takes into consideration our individual variation. And our individual variation evolved over a long period of time. And Rasmus is going to talk to us about how humans came from a past with certain climatic conditions, certain challenges in their environments, extreme environments, and how they adapted to those changes and those stresses. And Rasmus is one of the world leaders in this area. And let me just tell you a little bit about where he's been. The great thing about the interdisciplinary in this is that we're trying to generate a discussion and pollinate our different fields and come up with new ideas. But one of the challenges is that you won't necessarily appreciate the training that Rasmus comes here with, because he's been with some of the heavy hitters in the field. He trained with Monty Slatkin at Berkeley and got his PhD in the late 90s, and then he went to Harvard with a guy named Wakely, who is also a real heavy hitter in population genetics. Then he went to Cornell as an assistant professor, and then he was brought into the University of Copenhagen as a full professor, and then about 2008 moved back to Berkeley, where he's been since. So today, Rasmus is going to talk to us about, with the title, We're One, but We're Not the Same, the genetic basis of differential human physiology.
Thank you so much. I'm not quite sure how I get my my slides up there. Otherwise, I'm going to try with this thing here if there's nobody else here. I think it's regulated here somehow.
What if we just did you try?
I think it's a different presentation, so we need somehow to get rid of this presentation and I don't know how to do this. Maybe down. Yeah. No, this. Oh, that's the meeting. That's sharing the Zoom meeting. There you are. I can't figure out to get the presentation up.
Is there an escape button?
I tried to. So.
Which one is yours?
It's that one up there.
And then there's the F5 button.
All right. Thank you. Thank you. Thanks so much. All right. So, well, thank you so much for having me here, and it's a pleasure to be here. I think that sounds like a wonderful initiative. I think it brings together exactly the right kind of ideas. I think for the future in terms of improving precision medicine and thinking about wellness more generally. So my background is really in more in theoretical population genetics and in computational biology. I'm also by training a statistician, I'm also in the statistics department. But one of the things I'm been mostly interested in over the past 10 years is to try to use some of the ideas that we have been developing to try to understand human adaptation and evolution. And there are several different motivations for doing that. In human genetics right now, we know that there's a problem, and this is from a review paper from 2016 in Nature that point out this problem, that the vast majority of genetic studies in humans, of GWAS studies where people are trying to find associations between genetic variants and diseases, have been done in Europeans. It's becoming a little bit better. There are few more studies now, particularly of Asians. There are few studies including people of African descent, but it's really focusing on Europeans. It's a shame. It's a shame for two reasons. First of all, because those groups other groups than Europeans are being underserved. As we improve genomically informed precision medicine, people that are not of European descent, they will not have the same opportunities as other people. So it's a shame for that reason. It's also a shame for another reason, that is, we're losing out of an opportunity to really learn something about human genetics and human biology more generally. And why am I saying that? I'm saying that because humans, even though we're very similar to each other, are not quite the same because we've been living in somewhat different environments. Humans are very special in the sense that we are the only species on Earth that really have gone into all possible environments you can imagine. Everywhere except Antarctica, you'll find humans. One of the reasons why we can do that is, of course, because of all the technology we have invented that have enabled us to do that. But there might also be some biology to it. In some sense, in humans, we can't make experiments on people directly, but nature and human history has done that for us. And we can take advantage of those experiments. We can ask, what happens to people when they move into different environments? How have they adapted to those environments? And that principle has guided a lot of our research in the past 10 years or so, where we've been trying to look at different human groups and trying to understand how they adapted locally to their environment. So my passion is really evolutionary biology, and in evolutionary biology, most of the time the way things work is like this. We know something functionally, for example, about some genes, and then afterwards we're trying to say something about how they evolved. But I also argue that we can go the other way around. We can ask, okay, can we look at evolutionary processes and use that to infer function? So for example, can we find genes that have changed very rapidly and that can tell us what might be important for human fitness? And the principle here is that if there are certain genetic variants that are very, very important for our health, and where people differ by some people having one variant and other people having another variant of the same gene, then likely those variants are associated with both disease and fitness. Okay, so by looking at natural selection, you can get at what really is very important in terms of human health at the same time. And what I'm going to do here tonight is to give you some examples of that, of some studies where we have looked in different human populations to try to identify natural selection, where natural selection has been acting to change allele frequencies, and that way to get at what might be functional important for those populations. Although mostly what we're doing is we're really working on statistical methods. I know it's sort of in the evening here. You're all going to fall asleep if I talk a lot about statistical methods. So I'm not going to talk about that at all. But I'd be very happy to talk about it. It is really my passion. But What I'm going to do is just talk about some of the results that we've been obtaining. But the core of the methods we're using is we're really looking for populations compared to other populations where if we model the whole genetic variation, entire genome for all of the populations, where do we find genetic variants that stand out, that have changed frequency too rapidly in one population or another? That's the principle behind the methods that we're using. So the first example I'm going to talk about is Tibetan altitude adaptation. And let me start by giving you a little bit of background on that. So if you have ever been up in high altitude, you will know that you will then suffer from oxygen deprivation. That can be quite unpleasant in many ways. You can have some short-term symptoms of that, some more long-term symptoms of that. And you will then have an acclimatization response. That response will include an increased production of red blood cells. Why do you do that? Because if you have more red blood cells, you can better transport oxygen. So that makes sense. On the Tibetan plateau, the partial pressure of oxygen is only 60% of what it is at sea level. So you only get 60% as much oxygen with each breath of air. And one of the ways you react to that is by increasing the production of red blood cells. That sounds like a good idea, but it's not entirely a good idea for you to do that because it also increases the blood viscosity. Instead of having nice, good, well-flowing blood in your veins, you now have yogurt flowing through your veins. It's not good for you. It's not good for you for several different reasons. One of them is that it increases the risk of hypertension, ischemic stroke. There are also many complications associated with pregnancy for women that are not adapted to high altitude, but then live up in high altitude. So a lot of interest in what's in this problem. And what's particularly special about the Tibetans is that they don't have that reaction. In high altitude, they don't overproduce red blood cells. They produce about the same amount of red blood cells as they do in low altitude. So they don't have this maladaptive response to the hypoxia, the low oxygen levels at high altitude, as other populations group have. It's seen as an adaptation to high altitude. And what we're interested in is what is the genetic basis of this? Can we find genetic variants that can explain this difference between Tibetans and other people? And instead of doing a big GWAS study, what we decided to do was instead, let's try to find whether there's evidence of natural selection. That might take us there, because clearly this is associated with big, strong fitness differences. We know that Tibetans have adapted. So if instead of doing a GWAS study, we look at for where has there been natural selection, perhaps we can take a shortcut and in a much cheaper way get to the variants that are important. So we did that, and this is one illustration of the results. What it's basically supposed to show you is that there's one strong outlier, one gene that's called EPAS1. So what is EPAS1? It's a transcription factor, so it regulates what other genes they do, how they're being expressed, and it's particularly important in regulating the response to hypoxia, to low oxygen level. That was already known before our study. So it's a fantastic candidate gene. When you are exposed to low oxygen level, it's changes expression in the lung. When it changes expression in the lung, that initiates a cascade that leads to higher red blood cell production. Okay, so it's a good candidate gene. Another gene we found is a gene called EGLN1, and it sits in the same pathway. And it's also involved with the regulation of red blood cells. And about the same time as we published our study, there were a couple of other studies that also pointed all together to those same two genes, and particularly to this major effect gene, EPAS1. This this here is based on exome sequencing data, where we look at SNPs. So this is all just based on genetic data, no functional data at all. It's just a genetic data. So what we did was then to try to see, okay, is in fact the EPAS1 genotype that we're looking at, is it associated with red blood cell concentration or mean hemoglobin concentration, which is a proxy for red blood cell concentration. And what we could show this in a very little study, we just took a few hundred individuals, we could show that there was a strong decrease in mean hemoglobin concentration if you had the genotype that's presumably adaptive in the Tibetans. So we can explain, this gene alone explain about 40 to 50% of the difference in red blood cell count between Tibetans and other populations. Okay, so we think that by doing this, we found a genetic variant that is associated with the Tibetan high altitude adaptation, or at least that variant, or another variant in the gene that's in linkage disequilibrium with that variant. Next thing we did was that we were trying to see, okay, can we model how natural selection has been acting? We want to know, when did natural selection start to act? how strongly has natural selection been acting, and so on. And we started to do that. We ran into a problem. That was that none of the models that we used actually fit the data at all. They were all pretty bad. We could reject with strong statistical significance all of the models that we came up with, even when we made them quite complicated. And that is not something we had seen in other genes. So what was the problem? Well, the problem is illustrated in this figure here. And the way to read this figure is that you have the Han Chinese here. This is a reference population we compare the Tibetans to. It's the most common ethnic group in China. And then Tibetans up here. Each column is a particular SNP, a particular position in the genome, where if you are black, you have the derived type, the mutant type. If you're white, you have the ancestral type. And each row here is then a DNA sequence, and we only show the variable sites. And just looking at this, you can see that there's two very different type of DNA sequences in the Tibetans and the Han Chinese. There's one that's in majority in the Han Chinese and one in the Tibetans, and they're very different from each other. They are, in fact, too different from each other. For natural selection acting, one way you can think about it is, let's say that you have a new mutation, for example, it lands on a new chromosome, then natural selection will act to increase the frequency of that chromosome. Okay, but there's no reason why that chromosome should be highly, highly differentiated from other chromosomes in the population. You would not expect this degree of differentiation. So that was a bit of an enigma, a puzzle. We're trying to look at that. Then we have been part of the sequencing of the Neanderthal genome. So we thought, maybe it's because of one of these Neanderthal sequences that came into the human population that we now see in the Tibetans. Because we don't really see the sequence anywhere else in other people. We only see it in and around Tibet and the Himalayas. So we thought, okay, maybe that's it. Let's look at the Neanderthal sequence, which by then had been published, see if it's a consequence of this integration of DNA from Neanderthals into the lineage of modern humans that we now know happened. When we did that, we found that there was no match. So it's essentially there was very divergent from the Tibetan sequence, so that was kind of annoying. So we couldn't really figure out what's going on then. We have this really odd sequence in the Tibetans that's way too divergent, that's causing this re-regulation of red blood cells. What happened with that? And we put it away for a couple of years. We couldn't figure it out. But then, as Swantapavo was looking for more DNA from Neanderthals, the archaeologists he worked with, they said, this little finger bone here, that could maybe be a Neanderthal bone. Don't ask me how they know that, but they, it's sort of something about it. And so they said, okay, you can take that, you can grind it up, you can get DNA out of it, and see if that's a Neanderthal. Because Swantapavo, he kept searching more and more for Neanderthal DNA. And as he did that, it turned out that it was not Neanderthal. It was a distant cousin of the Neanderthals that we now call the Denisovans. So they diverged a long time ago from the Denisovans, or from the Neanderthals, and are quite genetically different from the Neanderthals. So we don't really know much about them, except we have their genomes, sequenced from just a few remains that have been found in the Denisova cave in south-central Siberia. So we compared to that sequence, and that was an almost complete match. So the Denisovan sequence was almost identical to the Tibetan sequence. So we think this is an example of adaptive integration. Adaptive integration means that you have a sequence that gives a fitness advantage. It's adaptive, it's integress, that means moved by interbreeding from one species to another species. And as we're learning more and more genomics and studying more and more genomes, we see that happens everywhere, all the time, between all kinds of species. And it's also happening in humans, apparently, quite rapidly. Now, to show that this is adaptive integration, you have to do a bit more than this. We make arguments using molecular clocks and recombination clocks and other sort of technical arguments that could show this is not just because we shared an ancestor with the Denisovans 600,000-700,000 years ago. This is in fact because it's been transferred by interbreeding. And I'm not going to drag you through all of those arguments, but one of the things we looked at is a haplotype network like this. And the way to read this figure is that each pie chart here is a haplotype, so a DNA sequence you find. And the color coding represents how many individuals in a particular population that have that particular haplotype. And then you connect haplotypes that are similar to each other, that don't differ by very many mutations, they get connected to each other. We have the Tibetans out here, and the Denisovan type here, and here we have the nearest other human haplotype. And the numbers here indicate how many mutations they differ by. So you can see there are 9 mutations between the Tibetan and the Denisovan haplotype. There are 40 between the Tibetan and the nearest other human haplotype. And then there are 35 mutations between the Denisovan and the nearest human haplotype. So in fact, the Denisovans are closer to other humans than the Tibetans are in that particular sequence. So you must say, why is that? Well, that's actually what you would expect if it's adaptive integration, because the Denisovan sequence is from ancient DNA. The individual that had that DNA sequence died maybe 50, 60,000 years ago. So that means that these individuals have had 50, 60,000 years more to accumulate new mutation. And if you try to model that, that fits very well with this difference in the number of mutations between the Denisovans and the nearest human sequence, and between Tibetans and the nearest human sequence. Okay, so that was the story of altitude adaptation in Tibet, where we could show that one of the key factors that allowed Tibetans to thrive at high altitude actually came from this process of adaptive integration from this other hominin species. What are other areas of the world where there's also been high altitude adaptation? We have two other areas. In the Andes, there are people in the Andes that have been claimed to have high altitude adaptation as well. And then also in Africa and Ethiopia, up in the Ethiopian plain, people have also been argued to have certain signs of high altitude adaptation there. So we looked at those two populations as well. So in Ethiopia, there was some already published data that we could do, where we could then compare some of these high altitude adapted populations, the Amharas in particular, to some other reference populations. And the gene that we found there when we did that was not EPAS1. It was in fact another gene called BHLHE41. The interesting thing about that gene is it interacts in the same pathway. So this is an interaction network. You have the central part of the network is really this protein here, HIF1 alpha. It's being regulated by EPAS1, and it's also being regulated by EGL1, which is the other gene I talked about. Another regulator of that gene is BLHE41. So what we think is going on in Ethiopia is there have never been Neanderthal, or Denisovans, sorry. There's never been Denisovans in Africa. So they did not have that genetic variant that selection could act on. Instead, selection acted on other genetic variability and another part of the same interaction network. And so the idea here is that you, at the phenotypic level, can get convergent evolution, but at the molecular level, modifying different proteins in the same interaction network. Okay. Last place in the world where people are claiming there's altitude adaptation is in the Andes, and we worked then in another project together with some researchers that worked on population in the Andes for a long time, particularly Joseph Precal here to the right, who really studies hematology and is a hematologist of training. And we looked at some samples there, and we came up with a list of genes, as we've been doing in the other studies, of which genes show most evidence of natural selection. And this gene was very different from the list of genes, was very different from the list we found for the Tibetans or for the Ethiopians. It's a quite different type of genes that we find. The second gene that we find is a gene that's already been associated with high altitude adaptation and has a well-known connection to hypoxia. The first and the third gene, the ones that shows the first and the third most evidence of natural selection, particularly this one shows very strong evidence. They're both associated with cardiac phenotypes. So atrial fibrillation, they're both in GWAS studies. They don't sit on the same chromosomes, but they're both associated with significant association to atrial fibrillation. So what does that mean? Well, we don't know it for sure, but it matches another observation done by an anthropologist, Cynthia Beale, who really established many of the early results in this field. And what she's been arguing for many years is that if you look in Ethiopia and Tibet, at high altitude, they have relatively low hemoglobin concentration, so low levels of red blood cells. So that's this compensation. They don't overcompensate by producing too many red blood cells. But something else is going on in the Andes. They don't do that. They, in fact, have the problems, the indigenous people in the Andes, that they do overproduce red blood cells. So she's been arguing that maybe selection has been acting on other phenotypes in the Andes. And what we hypothesize is going on is that in the Andes, for some reason that we don't quite understand, selection has instead been acting to mitigate the negative fitness consequences of having too high blood viscosity. And many of the mutations that we see that seem to be under selection, they're in fact in GWAS studies found to be protective against cardiovascular disease. So perhaps another solution to the overproduction of red blood cell is instead to try to fix the consequences of having too high blood viscosity. That's at least the hypothesis that we have right now. And there's a lot of research going on right now on people in the Andes to try to understand this better. So I think you'll see a lot of things coming out about this over the next few years. Another example that we're interested in are Inuit from Greenland. So Inuit are the indigenous people that live in Greenland. They also live other places in Canada and in Alaska. And they have been a group that anthropologists have been hypothesizing for a long time about having adaptations to the quite extreme lifestyle that they live on the edge of the ice. They're very highly specialized in a lifestyle where they live at the edge of the ice, hunting, fishing and hunting. They get a large proportion of their caloric intake from eating mammals, seals in particular, that are very, very rich in animal fat. And so it's been hypothesized that they could have different forms of adaptation. Of course, to cold itself. That's one possibility. And there's been many papers arguing that. But maybe also to this extreme diet that they have. There are not many vegetables growing there. So almost all of the food that they get is really protein and fat from fish and mammals. All right, so we did so I should give a little bit more background. So one of the reasons why that's particularly interesting is that they've been used as sort of a model to try to understand the effects of having a diet that's really, really rich on animal fats. So back in the 60s and 70s, there was a lot of awareness that it might be bad for you to eat too much animal fat. People started to question, well, what about the Inuit? In that traditional lifestyle, if we got to study them, they have a very high intake of animal fat. Some people say as high as 70 to 80% of their caloric intake is from animal fat. Some people have claimed in some studies. And still, on that diet, they don't seem to get a higher rate of cardiovascular disease than other populations. They seem, in fact, quite healthy, not struggling with obesity and cardiovascular disease on that diet. So how come that is? And then the argument went, well, it might be because of the type of fat that they're eating that is extremely rich in these omega-3 polyunsaturated fatty acids. Okay, so that was one of the major observations and arguments that led to the idea that omega-3s are protective against cardiovascular disease. So now, of course, if you go to the supermarket, that's a big deal. There are omega-3s everywhere. There are also papers that claim that if you give your children omega-3 supplements in the diet, they will become more intelligent. So all these American parents that are eager to make sure that their children get into Harvard, they make sure that they get omega-3 enriched milk for breakfast. So they will do well on the next test. So it's a big industry here. So we did a selection scan as we've done for the other populations. So that's another way to show the results is that we can go along the length of the genome, chromosome 1, chromosome 2, chromosome 3, and so on, and ask where in the chromosome do we find where in the genome do we find the strongest evidence of natural selection? In this case, for the Inuit, where do the Inuit have genetic variants that have changed allele frequency very rapidly, where the frequency of the mutation has changed in the population very rapidly? And we find a couple of interesting peaks here. This is the strongest peak and the second strongest. So now what we had to do, we had to learn about, okay, what does this might mean? Particularly, we get this very strong peak here around 3 genes, the FATS1, FATS2, and FATS3 genes. The FATS3 gene we don't know that much on, but the FATS1 and FATS2 gene is very well described, what the function is of those. Those 2 genes encode the proteins that catalyze the rate-limiting steps in the endogenous synthesis of these omega-3s and also omega-6 polyunsaturated fatty acids. So they engage in you can get these long-chain polyunsaturated fatty acids 2 different ways. You can get them through your diet, or you can synthesize them endogenously. And the genes that have been most under selection in the Inuit population are the genes that have to do with this endogenous production. And then together with collaborators, what we could do is we could look at the Inuit population and look at the fatty acids that were in the cell membranes of the red blood cells, and from that we could show that the effects of these of the mutations was in some sense to compensate for high dietary intake. So one of the enzymes, FATS1, the expression of that is reduced, and that leads to less accumulation of some of the polyunsaturated fatty acids that you otherwise would have gotten from, or tend to get mostly if you have a diet that's rich in meat or rich in fish. So it seems like what's going on is that there's a compensatory effect where, because on their traditional diet they would have this such high intake of those fatty acids, then genetic variants have compensated for that to re-regulate this so that the endogenous synthesis of those fatty acids had been lowered. Okay, so that's the proximal effect. But what does selection work on? Why is this so important for you? Well, it turns out that these mutations then are associated with a whole host of other phenotypes with extremely strong associations. So, for example, they are associated with LDL cholesterol. It's some of the strongest effect size on LDL cholesterol ever described in humans are by these genetic variants. But also other phenotypes, for example, weight. So of this lead mutation, the association with that, even if you correct for ancestry and other things, the effect on weight is 4 kilograms, right? That's a massive effect of just a single mutation. Average, it changes the weight by 4 kilograms. And then also this effect on many other things. For example, height is lowered by about 2 centimeters. So it's still one of the strongest effect mutations ever found, segregating at a high frequency in human populations. So massive effect sizes on many, many different phenotypes. So the height thing really has puzzled us for a long time, and the sort of story we came up with that was that we think it has to perhaps regulation of the release of human growth hormone. There's a lot of old literature going back to the 70s arguing that growth hormone release is regulated by the composition of fatty acid in the bloodstream. Chicken farmers, they put fish oil in the feed for chicken, and that somehow leads to increased release of growth hormone and the chicken become larger. Now, we don't know how this was work, and Ski was trying to convince me that maybe this is not the right mechanism, but there seems to be some association here with growth hormone and fatty acids composition, so it's possible it works like this. So our hypothesis on this is that we had this switch at some point in the ancestry of the Inuit, and it might go back much further than when they moved into Greenland. It's probably from when the ancestors lived in Siberia 15, 20,000 years ago that they started, where they switched to a diet that was rich in animal fat. They moved into areas where you really couldn't get much vegetable, or there were not many sources of food from vegetable sources, and you became highly dependent on diets that were rich in animal fat. Led to selection of these fat genes for one reason or another. And then they modified the entire fatty acid profile, has lots of consequences, reduced LDL cholesterol, also it reduces the fasting insulin levels. I didn't show that, but that's also true. Somehow that changes in the signaling or hormonal control and leads, for example, also to reduced height. The entire physiology somehow seems to be strongly affected by these mutations. That's what we think is going on. Certainly the strong effect sizes are right. The exact modeling of how it changes height, for example, we still don't know. But one thing we can say for sure is that the lessons from the Inuit regarding polyunsaturated fatty acids cannot be extrapolated to other populations. We're not all the same. Just because it's great for the Inuit to have a diet that's extremely rich in polyunsaturated fatty acids, long-chain polyunsaturated fatty acids, doesn't mean that's necessarily healthy for other populations. And there's right now lots of debate about this. How healthy is it really for you, for example, to take these supplements? And it's perhaps healthy for some people, but not for other people to do so. There's been large meta-studies of epidemiological evidence that argued that there's no protective effects of omega-3 supplementation in terms of protection against cardiovascular disease. And perhaps what the field is missing here is the genetic variability among humans, that we're not all the same. Perhaps it's good for some, but not for everybody. We're not all Inuit. All right. So, well, you ask about other populations. So before we published this paper about the Inuit, people already showed that there have been selection in these genes for some other variants, in these genes in Africa, and also for people in opposite direction moving out of Africa, already argued that it could be associated with diet. After we published our paper, there was another paper published showing that in South Asia, there seems to have been very strong selection on these genes associated with a switch to a more vegetarian diet. We are also interested in, well, what's going on in Europe? And so what we did in Europe was we took ancient DNA, that we have now plenty of ancient DNA, so for people from the Bronze Age, and then compare that to DNA today in modern humans. And then we asked the question, what are the genetic variants that have changed a lot allele frequency from the Bronze Age to now in Europe? And one of the top hits there, fat genes again. Okay, so this complicated figure that just shows various genetic variants and the change in allele frequency since the Bronze Age in Europe. Here you have the FATS1 and FATS2 genes. in Europeans, we can look at those variants, they also have strong effect on phenotypes, particularly on LDL cholesterol. It's not so clear that there's strong effect on many of these other phenotypes. They seem to be more specific to the genetic variants we see in the Inuit. But also in Europeans, we see very strong effects of these phenotypes on, for example, LDL cholesterol. And what we think is going on in the Europeans, our story about this is that at some point, that it varies depending on where you are in Europe, between 3,000 and 8,000 years ago, there was a switch to agriculture, from hunter-gathering to agriculture. That also implied a switch in diet, where instead of having a diet that was relatively rich in meat, you now had a diet based on cereals, based on grain. So much more vegetarian diet. That changed the profile in your diet of the different fatty acids, led to selection, to compensate for that, where you then have a haplotype different from the one in the Inuit, obviously kind of the opposite, that led to compensated in the Europeans and led to new metabolic changes in the Europeans. In Europeans, the allele frequencies of these variants are intermediate. So there's good reason to think that Europeans, depending on the ancestry and their luck, some of them might, for example, benefit from supplementation. Other people may not benefit from supplementation. But that's, as far as I know, not really still known. But we know at least that some of these variants in these genes that have been under very strong selection, they're nowhere near fixation. They haven't reached a frequency of 100%. They might be a frequency of 20, 30, 40, 50% in Europeans. So there's a lot to do there on trying to figure out what does this imply for recommendations, for example, regarding differential diet, omega-3 supplementation, and so on. Okay, so let me just for a second jump back now to the selection scan in the Inuit, because as I mentioned before, there were two peaks. There were in the fat genes, and then we had this other peak here. So this one here, we sort of know a lot of what's going on. We don't know the entire story, but at least we know a lot of what's going on with the fat genes. These other genes here, we don't really know what's going on. We haven't been able to figure that out. We still work on that, trying to figure that out. It's in a cluster near two genes, TBX15 and ROS2. Okay, so we think the selection is TBX15. It's an interesting gene. It's a transcription factor. It affects many, many, many different phenotypes, including skeletal development. But particularly interesting is it's also associated with differentiation of brown and white fat cells, brown and white adipocytes. Okay, so just to remind you, what are brown and white adipocytes? You have sort of the bad white fat. That's the fat you'll have around the abdomen. It stores fat. Okay, and the more you have of that, the worse you're off in terms of health. Okay, but then there's a good fat, the good brown fat. The brown fat is brown because it's filled with mitochondria that do a lot of respiration. You burn calories in them. So it's thought that the more you have of these, the better for you, because you'll burn calories and so on. It's also thought that the Inuits, several people argue, have more brown fat. I don't think that has really been established, but several people have been arguing that, and that's why we think it's quite interesting that this genetic variant, which is central in the differentiation of brown and white adipocytes, is the second most selected gene in the Inuit. But if it actually has to do with any of the distribution of brown fat and white fat, we don't really know for sure yet. We're still trying to work on that, trying to obtain biopsies from Inuit, where we can look at it. It turns out getting IRB approval for going around taking biopsies of people is kind of hard to get, we have learned. All right. Okay, so one little other note about the TBX-WAS2 integration. Even though we don't really know the functional story at all, we're just hypothesizing here based on what we know about the function of the gene. One thing we can say is that it looks like this is another example of adaptive integration. If we look at it, we have the haplotypes up here that are common in the Inuits, and these are the haplotypes we see in the rest of the world. This is the Neanderthal haplotype, and this is the Denisovan. And we can do various statistics to show that this kind of similarity, again, you wouldn't get that without integration. So we think we can show that the statistical evidence, again, this is a genetic variant that's been introduced into the ancestors of the Inuit from the Denisovans by interbreeding, that presumably, because of all the selection we see, have helped the Inuit to adapt to their environment. Similarly to what we saw with the EPAS1 and the Tibetans, although in this case, we're not quite sure what the function really is of that haplotype that's been integrated and how it has changed their physiology. Okay, but we think now that with these stories and stories coming out of other groups, generally that what we tend to see is that the more we look is that we see integration in many different parts of the world. And we think that there's part of the story of why have we been able to go everywhere? Why have we been able to adapt to all these different environments? A little part of the story, at least, is, well, there was somebody there before us. Okay, there were other hominins there that already had adapted to those environments. And we could go there and interbreed with them and that way pick up their genes that will allow us then to better fit into those environments. All right, so one more story here. I'm going to end with a story that we published last year about these very, very interesting people, the Bajau, sometimes called sea nomads, that live in Indonesia. They also live in Malaysia and in the Philippines. They also used to live in some places in China, actually. And they're very interesting for a number of different reasons. I'll get to that in a second. I'll just first introduce some of my collaborators. This is Eske Willerslev here, a long-time collaborator of mine who mostly works in ancient DNA. You see him here in his natural habitat in front of the cameras. And then also his student, Melissa Hilado, who really was the one who came up with this project. So she has been traveling a lot, and she was very interested in the culture of the Bajau, and she was very interested in perhaps they have some adaptations, and we could study that. Okay, so what's so special about them? Well, they live like, they're called sea nomads because they live on boats where they live over coral reefs and sail around from coral reef to coral reef, and then they have are known to have a lifestyle where they specialize in diving, where they can dive down to 70 meters, and perhaps more impressive that during their working hours, they spend 60% of the time underwater. So they spend more time underwater than above water. Each dive is typically not so long. It's maybe half a minute or so. They can dive for much longer time than that, but the typical dive is maybe half a minute. But then they just go up for maybe 20 seconds and then down again. Okay, and they do that for hours and hours and hours. The demands on your physiology from this, it's really extreme. Okay, you are exposing yourself over and over and over again all day, day after day, to acute hypoxia. Okay, so how do you adapt to that? Now, we don't know if there's been, we didn't know, at least before the study, well, they have had this lifestyle long enough to really show any genetic adaptations. Of course, evolution takes some time. All we know is that from the written literature, from other cultures have written about them, so we know they must have that culture for 1,000 years. But whether it's just 1,000 years or it's 20,000 years, we don't know that. Some linguists have argued that it's much older than 1,000 years, but we don't know because they don't really leave any archaeological evidence. You live on a boat on coral reefs, it really doesn't leave much archaeology. So we don't know much about the history in that sense, really, except that it must be 1,000 years old. Okay, so what can we study? What can we look at? What we're particularly interested in is in the diving reflex. And you might not all be familiar with the diving reflex, but it's a reflex that I think all mammals have. And it is induced by apnea, so by holding your breath, and by cold water immersion of your face. If you do that, that will elicit a specific physiological response. And that response includes bradycardia, so you're lowering your heart rate, peripheral vasoconstriction, so your blood goes to your central organ where it's needed the most, and also there will be a contraction of the spleen. Now, why do you contract the spleen? It's because the spleen contains a reservoir of oxygenated red blood cells. So when the spleen contracts, you get more oxygenated red blood cells into your bloodstream. So some people have argued that, it's like you have a diving tank with you. And if you look at certain other mammals, for example, the Weddell seal has a really, really large spleen, a very, very large spleen, and it's being thought to be an adaptation to diving, that it spends much of its time above water, but then when it needs to dive, it can then contract the spleen. And sort of a very clever way to avoid the problems that many people would have otherwise in high altitude, for example, of overproducing red blood cells. Now you can get all the red blood cells when you really need them, and the rest of the time you can store them in the spleen. And that's something that you see in many organisms also outside the diving response. For example, a horse, when it goes into gallop, it will also have a spleen contraction that can actually increase its red blood cell concentration by 50%, which is really crazy large amount. So it's very, very, very strong effect of these spleen contractions. Humans have it too, but maybe only increasing the red blood cell concentration by 5 to 10%. But that's still physiologically very important. Okay, so we really want to look at this diving reflex and the genetics of that. But there wasn't really anything known in the literature about it. This is the only paper we could find where people have done anything to study the genetics of the diving reflex. It's from this Russian paper where they study 4 candidate genes, and they argued that this gene, BDKRB2, is associated with differential diving response or diving reflex. It's the only paper that we could find in the literature that has looked at anything associated with the genetics of the diving reflex. I should say there's also very little in the literature about the genetics of the spleen. Apparently the spleen is not very interesting, so it's never included in any of the big studies, and there's not much information about that either. Now, what we're interested in the splenic contraction, though, and looking at that. And one thing that we could do very easily was measuring the size of the spleen, because that can be done with ultrasound scans. We couldn't get IRB approval for doing anything invasive. We could only get IRB approval to look at, to take a spit sample to do DNA, and then anything we could measure from the outside of the body. Okay, but one thing we could do would be to do ultrasound scans to measure the size of the spleen, because it's very non-invasive. Okay, so here's Melissa, the graduate student I talked about before. She's sitting in a village down in Indonesia and measuring the spleen as being entertainment here for the local population. As she's doing that, she was living in the village with people there for 2-3 months, and also did a lot of outreach while she was down doing this. And so this is one of her scans of the spleen. You're supposed to trust me when I tell you that there's a spleen here in this ultrasound scan. I actually don't know it myself, but there's standardized ways of measuring the size of the spleen, because there's certain pathologies associated with enlarged spleens. So there's standard ways of doing that, and she took a course in how to do this. And so what she did was she measured the spleen in the Bajau people, and then also to some other people living in Indonesia, the Salwans. And they're supposed to have a quite different lifestyle. They do agriculture, and they're never supposed to have had this diving lifestyle. And when she did that, she found that there's quite a variation in spleen size. That's not a surprise. But there are also, in average, much larger spleen size in the Bajau, much, much larger. And that could, of course, be caused by many different things. It could be just because you dive, for example, a plastic response, then the spleen tend to become enlarged by diving, or it could be because there's certain pathogens around near the Bajau villages that causes certain diseases that will make the spleen to become enlarged, and so on. Now, she did look at whether people in the Bajau villages right now that dive, and the ones that have stopped having that lifestyle, that don't dive anymore, whether they have differences in spleen size. And they don't seem to have differences in spleen size. So it seems to be at least not a plastic response to diving. But of course, many other things could be going on. But there could be a genetic effect. There could be some genetics involved in this. So we did what we did in the other for the other populations. We make one of these scans for selection, try to figure out where in the genome do you see most evidence of selection. We come up with a list, as we've done in the other cases, and it's presented here in terms of a list. And then we can ask, are there any of these genetic variants that are sort of our top hits, showing most evidence of selection, that are also associated with spleen size? And when we did that, we found one particular variant that is quite strongly associated with spleen size, given our relatively very modest sample sizes here of less than 100 individuals, and it is the gene called PDE10A. So what is PDE10A? Well, let me just show you this slide first. This just shows you along the length of the genome where you have evidence of selection here, and also there's a same area that's as evidence of an association with the spleen size. And then we could also show that there's expression variation here looking at, this is actually done in Europeans, but we could show the same region. We have expression variation that is associated with regulation of the expression of PDE10A. So it seems like there's been selection in this region, it's associated with the spleen size, and it has to do with the regulation of PDE10A, we think. Okay, so what does PDE10A do? Well, it's involved signal transduction, including adrenergic responses like the contraction of smooth muscles. Okay, we could then look at what other associations are there of these particular SNPs, and we find the strongest association we found was with hypothyroidism, so with levels of thyroid hormone. Now that's interesting because in mice, when you change thyroid hormone levels, then it changes the spleen size. Okay. And maybe before I go on, I should just say a little bit about ongoing research on what Melissa's doing for a postdoc. She's working in a mouse system on this, and she's been able to show that when you knock down this gene PDE10A, you actually get, when you modify the expression of PDE10A, you can get increased spleen size experimentally in the mouse system. So it seems like it's in fact true that PDE10A has effect on the spleen size, also in an experimental system. We should also say that we could go to other cohorts. Nobody's ever done a GWAS study, a genetic study associated with spleen size. But we could do it a little bit ourselves by collaborating with other people that had abdominal scans done together with genetic data. And we could show in a European cohort there was also association in a European cohort, marginal association with these variants and spleen size. But the variant is a very low frequency in Europe, so we are very underpowered to do that. Okay, so what we think we can conclude from that is, first of all, there's been selection leading to increased spleen size. We think that we can make that argument, but then we're wondering, okay, what about all these other genes? This is, in fact, not the gene that shows the strongest evidence of selection. Well, what is the strongest one? That is this gene here. So right out upstream of this gene that they have found was associated with the diving reflex. That's where we find the strongest evidence of selection. What does that mean? We have no idea. We don't know. We don't have any phenotypes right now where we can look at that. We're right now working on going back, getting IRB approval to do the things that we need to do to try to look at what might be the effects of these genetic variants. There seem to be a very strong selection on them. They seem to be associated with the diving reflex, diving response. But how the diving response has been modified in the Badger people, we have no idea about that. All right, so conclusions. There's been selection on PD10A that we think has resulted in increased spleen size. And then there's also some selection on this BDKRB2 gene that likely has modified the response to the diving reflex. We think, we hypothesize. And there's much more work, obviously, to be done on this system, particularly really to understand the physiology, which is really very undescribed. Not much work has been done on that. So working together with several different physiologists really to try to understand the physiology much, much better, also before we connect it to the genetics. All right, so with that, I'd like to end. I'd like to thank all the study participants that's been part of the studies I've been talking about, and then all my many collaborators. And I want to make sure to mention some of the younger people that really have been leading much of this research, particularly Lisa Lado, who's leading the research on the Bajau, Emilia Huerta Sanchez, who was leading much of the research on the Tibetans, and also Ida Malke and Matteo Fumagalli and Hans Albrechtsen, who did much of the work on the Inuit. And of course, thank you so much for having me here today. It's been a pleasure to talk to people, and thank you for listening to me.
Okay, we have time for a few questions for our wine break, and then we're going to come back and have a panel. Pardon? Yeah, we'll
Really
have
have questions.
help open it up for more questions at the end. But any immediate questions?
Yeah, so I mean, so any sex-specific effects? Yeah, so often in these models we stratify by sex, so it's sort of covariate which sex you are. So it's in the statistical models. And for example, for something like the spleen size, there's a very big difference between males and females. Have there been differential adaptation where it's the males that have adapted, not the females? We don't really see evidence of that strongly, but it can be quite hard to show when the base when there's already a big difference between them. But for example, for the Tibetans, there's also a difference in hemoglobin concentration between males and females. And why there is that, I'm not quite sure. Yeah, but that's also true. Yes, good question.
Question? Hang on.
Very interesting studies and very clever approaches to using both the genetics and clinical phenotypes in order to reduce, I think, your population size. Could you provide us a bit more information as how small the population you need as compared to a conventional GWAS for the same effect size, and also which mathematics you've conceived around that?
So a little bit about the sample size. In several of these studies, it was about 50 and 50. So orders of magnitude less than what we would need in a GWAS. And then because all we need to find is just where has there been a change in the allele frequency, really. And you don't need that strong. If there's been really strong selection, you can get away with small sample size. So we can do this study at a fraction of the cost of a GWAS study. We have used, in terms of the mathematical methods, we're doing many different methods. Right now, what we're doing is sort of a complicated methods where we model the population's genetic structure similar to a model like the one that you have in programs like structure and mixture. So you assume that each individual fractionally belongs to a population, and we estimate the allele frequency and the fraction of each individual that belong to that population group with maximum likelihood. Together with that, we also estimate a tree of the history of those populations. Then we model the change in allele frequency as a Brownian process motion along the edges of these trees. And then we can establish like a ratio test that tests whether there's more evidence of another factor that changed allele frequency faster than predicted by the Brownian motion process on particular lineages in particular SNPs. So that was a short overview of that. Well, we also use simulations, yeah, lots of simulations. Well, simulations typically, so there's some fundamental problems in population genetics. Now I'm gonna geek out a little bit now on these issues. But in population genetics, we have sort of challenges that making statements about selection, that depends, you can't really make that without having an idea about the background demography. That is the history of the populations. Now, to deal with that, we model that history. That's why we have these complicated statistical models. Even doing so, that we might have failed occasionally. To get statistical significance, we typically use simulations under very complex models to try to get a statistical significance. But mostly what we're doing is that we're modeling the joint distribution of allele frequency in the entire genome for all individuals in all populations first, and make a model for that. And then in some sense, we find outliers for that model. That's another way to think about it.
Okay, I think I can. Okay, so the spleen is also an immunological organ. Have you tried, or somebody is trying to find out whether inflammation is different in these populations? Because this is one of the very important problems in.
Yeah, absolutely.
Yes.
So that's a very important question, and we really, I don't have any more data about anything than what I talked about here, and it all has to do with getting IRB approval. So for example, we did not have IRB approval to take a blood sample. So you have to do everything you can do without taking a blood sample. That's not very much. So that all we have is that. We're trying to get exactly to answer those kind of questions and also look at immunology. We're trying to now get IRB approval to actually be able to do that. Good question. Yeah. I should say, why is it difficult? These are indigenous people, right? So you don't go out to indigenous people and just sort of like sample them and so on. I mean, there's a whole different process that's really important when you work with indigenous people, of involving the community and so on. We have to show that we're really doing that in a responsible way and that the indigenous community gets something out of it. That's why the IRB process is more challenging. Anyway, go ahead.
So thank you for your talk. How can we take this, this examples or these cases and apply them to clinical medicine, given the topic of precision wellness, and how to really take the ideas and concepts here and bring that to health in human disease?
So in several different ways. I think the FATS genes is a great example of that, and you have one of the world's expert on those and the physiology of that sitting right in the front row. The question is, how can we immediately? I think these results suggest that we have some differential physiology in response to diet, and it might you need to do a bit more studies, but very soon to recommend differential diet based on genetics. I think the FATS genes are some of the most promising at all for doing that, and I don't think that's very far away. Ski and other people are going to do that and solve that problem for us very soon.
Yeah, let me say something about that. I mean, in Africa, the FAGS gene, the frequency of the efficient variants are around 83, 84%. In Caucasians, that's about 43%. So in 1961, the American Heart Association said, eat saturated fats are bad, eat PUFAs. We instantly went from 1% of our energy, omega-6 PUFAs, to 8% of our energy. So we dramatically changed exposure. Now you have 8% of your energy as linoleic acid, the omega-6, and African ancestry populations can take that to arachidonic acid and several hundred pro-inflammatory messengers much more efficiently than Caucasians. That's a pathogenetic mechanism that can drive health disparities, and we continue to do that in every areas. But what Rasmus is doing is amazing, and once we understand frequency differences, once we understand we're not all the same, and we understand how, and then we're looking at exposures to nutrients or any other environment, that becomes very different with very different health outcomes. I think we're going to have a wonderful discussion here. If we'll get 15 minutes or so with the wine and cheese, I know there may be other questions, but hang around till after the wine and cheese, because 4 of them are going to go at it, and we're going to go at them, and we're going to have a good time. So this is what science is all about. It doesn't get any better than this. So go get some wine and cheese.
me very nervous with you know, you sitting in the audience knowing those pathways so much. But, okay, but I went back to myself, like, okay, I'm gonna.
stand. Hello, hello. Do you want to explain what the
I will. I know. I'm just going to say what the point of this is as soon as we get a little bit more people in the room. I think we'll get started. This is the fun part. Rasmus gave a fantastic talk, and rather than just walk out and say that was a great talk, and a few weeks later forget about it, we thought we'd use this as an opportunity to stimulate conversation across departments with the idea, hopefully, perhaps somebody's going to get a great idea for collaboration to really bring this interdisciplinary group together and put our heads together. So what we have at the end of each of these seminars is a panel discussion, and it's all local folks, and their work doesn't necessarily tie into the speaker. That's the point, is to introduce more diversity to the evening. So we have three panelists tonight, Brian, Joanne, and Ryan. They will introduce themselves, and each of their work ties in some ways and doesn't in other ways, and you'll see that it'll make an interesting combination, a network of ideas. So if you want to start, Brian. A couple of slides to...
Yeah, so this will just be a short little overview, just so that you know who we are and what we're working on. I'm Brian Hallmark. I'm a researcher here with Bio5. Can you all hear me? And so over the last few years, we've been working on something very similar to what Rasmus discussed today, and this is a subject very dear to my heart, which is looking for evidence of genetic adaptation to climate and diet in Siberians. These are some of the folks that we've worked with over the years. Now, as you probably know, Siberia is really cold, and as modern humans left Africa and entered Eurasia, they found a much colder climate. In Siberia, they also found a very limited diet and months of near to total darkness. But there's archaeological evidence of settlement of 72 degrees north by 45,000 years ago, even though we don't necessarily think the modern Siberians necessarily descend from those original inhabitants. They have been there for a very long time. And so just, I don't know if you can see the scale, this is negative 48 Celsius, so that's the average January temperature in this region. I was going to put the temperature from today in Yakutsk, the coldest city, but they were in a warm spell and it was 10 degrees today, so I didn't think it was very compelling. But so as Rasmus mentioned, you know, just because something seems like it might be a selection pressure doesn't necessarily mean that it is. And humans, as they expanded around the globe, developed many technological and cultural devices to cope with difficult environments. And so even though it's really cold, it's not necessarily clear that negative 40 degrees is worse than negative 20 degrees or 0 degrees, or, you know, well, the freezing point of water is important. But in other words, there are cultural and technological buffers to these extreme environments that, and there's also phenotypic plasticity that allows you to alter your own physiology given a new environment. So we were interested in, and let me back up and just say that we, in this particular project going forward, did not have phenotypic data. So we were using what's been reported in the literature, and it's known in Siberians that they have an elevated basal metabolic rate. They have higher thyroid hormone, especially in winter, though that varies in non-Siberians as well. And it appears that they may have some physiological adaptations that may have a genetic basis. So we weren't going into this blind, but we were doing selection scans similar to the types that were described previously. And just in terms of what our data, we had both whole exomes and high-density SNP array data, for those who know what that is. And our goal really was to reconstruct demographic history. There was a mention that you need to understand demographic history without selection to make inference about selection. And so we also wanted to understand adaptation to climate and diet. So I should say we focused on just 2 populations with our exome data, but then we had many, many different Siberian populations, and I do not mean to imply that all Siberian groups are the same. They are not, but for here, I'm kind of lumping them together. So in terms of results, which I don't have time to discuss in depth, I just want to say we have a number of publications out already on this, including the very complex history of Siberia. But more recently, we've found some interesting results and patterns indicative of selection. And one of the papers is this one over here on exome sequencing. And in this paper, we found what looks like we didn't find any really strong signals of selection like some of the ones other people have found, for example, in Tibet. But what we did find is signals of what would be called polygenic adaptation, which is signals on kind of gene sets, or in our case, biological pathways that we believe are under selection due to perhaps cold or a fat-rich animal diet. Lastly, and more recently, recently published this paper in which we found evidence of selection at 3 loci. All 3 are involved in lipid metabolism at a very fundamental level, and all 3 are highly related to brown adipose tissue function. All 3 are missense mutations, and I could go on and on about these, but I will spare you. The main point is just that we're finding some interesting results here as well. The last thing I wanted to mention is that in the Inuit study that Rasmus discussed, the second highest peak was in this TBX15 Wars 2 region. And here's the paper that he discussed, and that it comes from archaic introgression. So we were interested in what this looked like in Siberia, and so we genotyped SNPs across this region, and not only do we find a quite dramatic climb into northeastern Siberia, and then find it nearly fixed in the Americas. These are admixed 1,000 genomes populations. We find that the introgressed haplotype is on a much longer haplotype, and that haplotype has some very, very interesting geographic patterns, including being found in Europe. And right now we're doing some more work to look at ancient DNA to try and understand when this haplotype may have introgressed. So the search goes on. I just want to acknowledge especially Tanya. She has been my partner in crime. She is now retired, and she is the Siberian scientist who collected most of our data. And I want to also thank all of you.
I'm going to jump right into the next. Success. So my name's Joanne Burkout. I'm a research assistant professor in the Center for Biomedical Informatics and Biostatistics here at the university. I'll give you a little departure from climate and from cold and talk about the genetic legacy of infectious disease and its consequences for valley fever, which is a project that we're working on right here. So it's no surprise, especially given the introduction, if you've been here for the whole thing, people respond differently to everything. People respond differently to infection. And a lot of that, infection is a very complex response. It involves the human host responding to the pathogen, doing a lot of compensation on their path from disease into health or recovery, trying to balance host damage and other trade-offs along with pathogen control. And there's not a one fixed best way to respond to infection writ large. So different populations in the world have really undergone different very severe either steady selective pressure, such as malaria and tuberculosis, which kill millions of people per year and have over the course of human history killed probably billions of people. There are also epidemics and pandemics like cholera, like bubonic plague, like Spanish flu that act as a very strong selective sweep and can kill off millions of people in the span of a couple of years, leaving a set of people in their wake who may or may not be a random set of what started. And we've got a lot of research in the field looking at a lot of these different things, looking from the perspective of specific pathogens, and we can find a lot of signals of genetic selection on specific genes, specific pathways that are relevant to specific diseases. Some of these trade-offs, some of these have cross-reactivity and cross-activity across different diseases. Many of them do not. But this is more or less just to tie up into the previous set of talks and just say that we can't forget that even though we're living here in the Western society right now, infectious disease has been a huge part of human history. It's absolutely shaped our genome in many ways. So now I'll move into valley fever specifically. So this is the distribution of valley fever you see on the map. Tucson is an epicenter for it, but it is still a very rare, very uncommon fungal infection. It's caused by fungus that lives in the soil. It's inhaled, and then what you get, and you can see this little Sankey diagram here, which don't read the numbers too closely, but the proportions and percentages should be representative of what we see. There's a lot of people who are exposed. The overwhelming majority, even if skin test positive, are asymptomatic. They just don't develop disease in response to exposure. There's a small subset that do develop clinical presentation, and a small subset of those patients, the majority develop a pulmonary self-limited disease. Basically, they get pneumonia. They can clear it, they can resolve it, they can be treated for disease, but there's this very small percentage which gets disseminated disease or extrapulmonary infection, where the fungus can escape from the lungs, where it was originally inhaled, and begin colonizing in the skin, in the brain, in the bone. And this is unfortunately a lifelong problem for those patients which have dissemination. They aren't able to clear it, they need to be on lifelong therapy. So this is really where we want to focus. Why is it that there are some patients who disseminate while other patients do not? And we have good evidence that there is a genetic trait. There's a genetic basis behind this trait. Black Americans have 14-fold higher rates of dissemination relative to white Caucasians or white Americans. People with Asian ancestry have higher, but not quite as high as African American. We have good evidence from the occasional Mendelian case where we do see immunodeficiencies, and we can see a lot of these things that hold even when exposure is controlled. So looking at, say, a dust storm that swept through, or exposure in a prison context where the people really are all going to be exposed to the same exposure. So why is it that there are some people who develop this severe response versus those who are able to limit it a little bit more effectively? So the complications of studying a disease like this are not very straightforward. One, it is rare. We are looking at handfuls of patients infected per year, handfuls of patients who are disseminated. So accruing a sufficiently well-powered and unconfounded cohort to conduct a case control study as you would do traditionally, it's just impractical. We would love to do it. We can't really practically do it. It's mostly complex inheritance. So I did mention we have Mendelian cases, but mostly what you're seeing is genetically complex. So we're not looking for one signal. We're looking for a bunch of different responses that sort of converge into common pathways is our expectation, very heterogeneous. And it is a dynamic response phenotype. So we can't ethically go in and just infect 1,000 people and find out who's disseminated and is stuck with this disease for life and try to figure out from there what we're gonna do. So we need that exposure to happen naturally in an observational context. And the signal may actually be obscured within the resting state because we want to know what's the problem with dissemination, not what's the problem or genetic factor that influences exposure in the first place. So it needs a provocation. The approach that we're using, it's called an N of 1 pathways gene expression profiling approach. It's a single subject method. And so there's a component of a bioassay and then an analysis method. What we do is we take some of these pulmonary or disseminated cases who are brought here because we've got the Valley Fever Center for Excellence right here at the University of Arizona. We take their blood, we put it into a dish and culture it. We take one aliquot of blood, leave it unstimulated. We take a second aliquot of blood and stimulate it with coccidiomycosis lysate, spheral lysate. And then we collect RNA sequencing data from both of those pools, and we look to see what are their transcriptional response to stimulation, which we hope is a model of what is the response in their human host body to identify disease. So, like I said, it is complex, and we have statistically a pretty significant problem with multiple testing and heterogeneity. So what we do is we take that expression, we look for those genes that appear to be differentially expressed in response to infection, and we try to condense them into biological processes and look for overrepresentation, which can reveal sort of more higher level and systems properties of the response that this specific patient is having. Our goals are many. We have a biological goal, which is to understand the host response to cocci. This is a rare and understudied disease. We want to identify response factors in these patients that differentiate those with dissemination versus those with self-limiting pulmonary disease. We work in the Center for Biomedical Informatics and Biostatistics. We want to develop the statistical methods and improve those to accurately and meaningfully describe single patient paired data. And we're looking to develop statistically and biologically sound ways of comparing two or more groups of patients on the basis of their systems properties. And that's something we're working on right now. So I'm not going to show you a lot of results right now. It is a work very much in progress. I do want to acknowledge the other people who've worked with me on this. A lot of this work was done in the group of Yves Lucier, specifically Chika Lee and Nima Pulati have done a lot of work in actually analyzing these data. Also Haichuan, Walt, and Helen with developing the statistics. Valley Fever Center for Excellence has been our collaborator in this, primarily John Galgiani. Jeff Frellinger, Dan Powell has been doing all of the bioassays, so we absolutely appreciate his hands, and a lot of this is funded through a UO1 grant. We've got our partners at the NIH, who's Steve Holland and Amy Hsu. While I have the podium, I'm also going to throw in a little plug for next month. We've got a Women in Data Science Tucson event happening next month, April 5th. If you'd like to come join us, and if you're using big data analysis, if you're developing big data methods, come and see what we're up to. If you are a woman doing these things, come and present with us. We'd love to see you. So find me afterwards, follow that link, and pass over the mic.
Okay, so I'm Ryan Gutenkunst. I come from the Department of Molecular and Cellular Biology down on South Campus, where I'm a faculty member. And Rasmus told you that he was going to avoid talking about the sort of nerdy pop gen side of what he does. That's most of what my group does, so I'm not going to spare you that from now on. And in fact, we're best known in my group, I would say, for working on building models of population history that sort of serve as the null models for a lot of the statistical stands for selection that Rasmus talked about today. But I just want to show you one project that we're actively working on in our lab, and that has to do with not adaptive variation, but thinking about the whole distribution of fitness effects. So the adaptive mutations and variation that Rasmus was talking about in his talk is really the tip of the iceberg over here. And as you might expect, most of the mutations, most of the genetic variation that we see in humans is not adaptive and beneficial. Much of it is evolving roughly neutrally. And then there's a large proportion of variation that is deleterious. Some moderately deleterious, some very strongly deleterious. And the shape of this distribution of what are the effects of new mutations on the fitness of the organism is very important for determining the evolutionary and population genetic properties of any evolving population, be it humans or Drosophila or a pathogen. And so for many years, people have been trying to learn about this distribution. It turns out to be difficult to measure experimentally because, particularly to narrow in on this region where things are slightly deleterious, because the effects of these mutations might be very important over evolutionary timescales, but difficult to measure in the lab. And theory also doesn't tell us a whole lot about what this distribution looks like. So to learn about it, what we and other groups are doing is going to contemporary genetic data and then modeling the variation we see in populations nowadays and asking what shapes of this distribution could have produced the patterns of genetic variation we see nowadays. And so my group, we focus on developing new population genetic methods and developing models that then we then apply to data to learn something about population genetic questions. And today we're trying to learn more about the shape of this distribution. And the innovative thing that we're doing in our group is that we're trying to generalize this idea of a distribution of fitness effects to think about not just a single population, but to think about multiple related populations. Because many of the species we're interested in are localized not within a single point on Earth, but instead have expanded from some ancestral homeland, perhaps. And as they've expanded across the globe, these populations have encountered new environments. And any given mutation, if these populations are recently diverged, might appear in both populations, but it might be doing different things in those populations. So in the ancestral population, it might have a certain effect on fitness, but maybe in this new environment, it has a different effect on fitness. And we'd like to understand on a very broad genome-wide scale, what do we expect? What sorts of differences can we expect to see when we look at fitness effects of mutations in different populations? And so to do that, we need to go from thinking about a one-dimensional distribution that exists within each population to think about the relationship between them. How are the selection coefficients in population 1 related to the selection coefficients in population 2? And I won't nerd out too much, but I will say we have a couple, just to give you some intuition, there are a few extreme possibilities. One might be that we see a distribution like this that is very highly correlated, such that basically all the mutations are having the same effects on fitness no matter which population you're in. You mess up the organism in, say, Africa, you're going to mess it up just as much in Europe. At the other extreme, we might imagine a distribution that is very wide, where this subset of mutations is doing very different things in the two populations. Maybe these are mutations in very environmentally responsive genes and might be interesting when we think about local adaptation. And so we're interested in this because we think it's a fundamental population genetic quantity that has not been explored, and that it'll tell us a lot about speciation. So how do new species form when populations diverge from each other? Digging in a little deeper, we want to use this as a window into thinking about how mutations in genes interact with the environment and interact with other mutations, other genetic variants that are segregating within a population. And to get to sort of a practical human level, we think, as Rasmus mentioned, that most genome-wide association studies are done in Europe, and people try to transfer those results to other populations around the world. We think that this fundamental population genetic quantity about how mutation fitness effects differ between populations will play an important role in understanding how much we can expect to transfer results from GWAS from population to population. So I won't get into the details about how we do this. I just want to very briefly talk about some preliminary results from a couple applications. So first, we looked at Drosophila because it's one of the most accessible population genetic model organisms. And being a computational lab, it was very convenient to just go out and download the data. And if we compare African flies and European flies, we find that this distribution of fitness effects is very highly correlated, with a correlation coefficient of about 0.97. So most mutations do very similar things in these two populations. But if we break our analysis down to look at genes of different functional categories, we see interesting patterns of differences. So for example, the biggest differences we see between populations are in genes involved in immune function, where there's still a quite high correlation, but is much lower. And if you know something about evolutionary biology, you might think, well, that's not surprising that as Drosophila has expanded across the globe, it's now encountering new pathogens. And some pathways are more important than other pathways. And so mutations that affect one pathway will have different selective effects in Europe and Africa. So it's a very natural biological interpretation of this result. We've also done some preliminary studies looking at humans, taking advantage of the 1,000 genomes data. And so far here, to be frank, the story is not that exciting. So between Africa and Europe, we find that the correlation of the DFE is indistinguishable from 1, and that there's little variation among gene functions. And this to us actually raises some interesting questions. So why don't we see the same effects that we see in Drosophila in humans? And there are a couple of very natural hypotheses. One might be that humans are less genetically diverse. The other might be that, of course, we control our environment. So the populations we were able to access in the 1,000 genomes are agriculturalists in Africa, and of course agriculturalists in Europe. So even though they're living in different external environments, they've controlled their environment quite strongly, so that in many ways they're living similar lifestyles. We're interested in extending this analysis to think about, say, agricultural Europeans versus hunter-gatherer Africans, where we might expect to see bigger differences from populations that are exposed to different challenges. So I'm interested in this sort of nerdy world of population genetics theory and trying to get at the fundamental population genetic and evolutionary processes that maybe take a few steps to get us toward impacting human health. But I think accessing these fundamental questions will really help drive us forward in the long run. Thank you.
So we can take questions for any of the presenters. And great ways to synthesize. To carbonated drinks. Something
That might be a lot to ask for, the carbonated drinks. I don't think I have any particular deep insights on our ability to adapt in the future. It has to do with how much genetic variation there is. I mean, the question people often ask is, are we still evolving? And I think to that, we must be. I mean, there's still, some people have more offspring than other people, and that does correlate with something genetically, but maybe in a quite different way than what we think. So we are perhaps evolving, but not maybe in the way that many people would think that we are evolving. Well, we can adapt, and is it even relevant if we can adapt? We can just modify ourselves, right? I mean, we modify our environment. Maybe we are evolving to have much poor eyesight because we accumulate all these deleterious mutations because we have glasses, right? I'm in the situation now, I can't see anything on the screens because I'm in denial, I need glasses, and so on. And so, other people in this room that is getting into my age and so on, you have the same problem. But we have invented glasses, right? So I think maybe the idea is not that we need to adapt, but we just, have technological fixes for all the problems that we create for ourselves.
I don't want to dominate, but I've got a question for everyone. So if anyone has questions, interrupt me. Let me start with Joanne. So valley fever.
Valley fever.
14
Welcome
times.
to Arizona.
Yeah, but 14 times more prevalent in African ancestry populations.
Yeah,
African Americans.
It's absolutely huge.
So
effect size.
I was looking at the genes, and they're some of my favorite because we study inflammation, and I was certainly TLR, the endotoxin receptor genes, NF-kappa B, the central regulator of inflammation, First of all, my question to you is why do we see this map that, and again, I'm dumb as a shoe in this area, so why do we see this map that is only in the Southwest, and we're not seeing it, in African American containing areas if it's 14 times more prevalent? And the second question is really, these are some of my favorite genes. We're right now looking at lipidomic and metabolomic analysis of healthy humans that were stimulated with endotoxin and LPS. So we're very interested in those receptors. So, does that say something, does the 14 times more prevalence say something about these particular genes and their response to their inflammatory response?
Right, so here's your first question first. It looks as though, and right now we're a little bit too preliminary in our recruitment and analysis to give a strong answer on the complex side of things. Thank you. But when we look at monogenic cases, people with primary immunodeficiencies, it really reveals that valley fever seems to be related to, JAK-STAT, IL-12, that particularly inflammatory loop. And that particular inflammatory loop is actually really, really important for tuberculosis. So we do not expect that valley fever has itself exerted much of an impact on people with African ancestry. Valley fever is in the Southwest. People with African ancestry who come to the Southwest are all imports, at least evolutionarily speaking. So valley fever is not the reason that we would see these changes, but the fact that ancestrally and evolutionarily speaking, people have been exposed to other pathogens that have influenced the way that they then respond to a new pathogen, I think is really an interesting story. Like I said, I have my personal bias is that I think tuberculosis is a major player in this. This just comes from my own background. That's what I did my PhD in. And I recognize all those genes too. And yeah, so there's definitely something complex happening as well. We're not going to find a single gene signal, but we're looking at the way that some of these populations in Africa seem to be responding is very detrimental in this environment, and we'd love to know more about it. I'm just gonna get your second question before I'll take your question before I move to the second.
Sorry. I guess maybe I missed it.
So that's an excellent question that unfortunately has not been addressed in the literature that I've been able to see. So it doesn't seem as though people with Native American ancestry have particularly high rates of cocci, but people who don't show up to the hospital are a little bit difficult to do surveillance on. So whether they're just at home suffering and physicians never see them, or they're truly more resistant, or it's just a numbers problem, I don't know what the answer is.
How bad is it for African? So say I have a friend who wants to move here and likes going outdoors. Like, what is their probability of serious disease? And should I actually counsel them not to come?
It's still really rare. The fungus is everywhere. Exposure does not seem to have a racial or demographic divergence. Seroconversion doesn't seem to have a racial or demographic divergence. It's just in this small percentage, and we're talking 17,000 people a year in the entirety of the United States, the entirety of Arizona and California combined. So, tell your friend not to go out in a dust storm. If your friend is a serious gardener, there might be a slightly higher risk. If they do develop a pneumonia, tell them to consider valley fever. The physicians here definitely know what it is. But we're talking, it's a rare disease. So I wouldn't, at this point, tell people to stay away.
I have another question, but maybe answer it later. I want to be able to
Yeah, I think that's a really important consideration. Unfortunately, I don't have an answer for you. I'm a geneticist rather than the person out there in the clinic. I would love to have an answer. If you want to talk after, we can think about it, but I don't know. I think it's a great point, though.
I consider what I heard today, and they are the same apparently, and yet it seems to me that the way to introduce those genes into the general population is to allow or somehow encourage these outliers to have more children with the general population, because I don't see how can we get any other way the advantage of these new adaptations. Do you have something to comment?
Briefly to address that, I think the sort of applications of this is not so much that there's certain variants that might be beneficial that we should spread among other people, but it's more that what we have learned about how the genetics affects the physiology. And just for example, that if there's differential genetics that determine differential physiology, we might know something about diet and give people better recommendation about that. We know much better about interventions. We know, for example, for the acute hypoxia in the Bajau people that I was talking about, the divers, people that are doing emergency medicine, they're very interested in acute care of people because what often happens when you are in trauma is that you have hypoxia to the brain, and that is really the long-term effects is the trauma to the brain. Now we have a system where we can learn about what might determine which people perform better or worse, or have better or worse outcomes depending on the genetics when they're subject to acute hypoxia. So it's more those kind of things we learn rather than I think that we want to introduce the variants into other people. I don't think that that's what we're trying. I know there was there's one experiment in Shenzhen in China, right, where they're trying to do that. I'm not sure that that's what we're advocating here.
So I get, well, I can tell. So I'm going to ask Rasmus questions at dinner, and Brian, he's in my lab, so I'll pick on Ryan here. So your models, and again, I'm just poor country boy here, but your models are so when you so we're looking at the FADS variants, and we're looking at this specific gene, and we've now just looked at the association of 4,000 variants in the FADS cluster and a metabolomic. So we're looking at 600 small molecules, and we're seeing these incredible differences not only in frequency of the FADS variants between Africa and Europe, but that translates into molecular phenotypes that have real effects on human health and disease. So I mean, I guess I don't when you say we're not seeing differences between Africa and Europe, and I understand you're looking at whole diversity, but when we're looking in the weeds, something that is crucial, a gene that evolutionarily went up and down, up and down, up and down. Every time you went to a vegetarian diet, or you went to this, or you went into the cold, or you came over, so the frequency difference between these variants are they're 40% between African and European ancestors. So again, I don't understand the models, but I guess my question is, are the weeds more important than the whole, and are you looking at the whole, and because when you're looking at the gene diet interactions we're looking at, they're crucial to human health.
Yeah, I think that's a question that's both very specific and very broad at the same time. I think it's an open question, I would say, in sort of the population genetics and evolutionary biology literature, at least in terms of thinking of human adaptations. are the sort of single gene stories like Rasmus has told us tonight, is that the typical mode of human adaptation, or is it the sort of polygenic style that Brian mentioned, where it's not a single hot gene or hot variant that is going up and down very dramatically in frequency, but instead it's subtle changes in lots of genes that are sort of accumulating in a population? And I think Rasmus may disagree with this, but in some sense, the signals he's seen when you have these single gene hits are just, they're like blazing beacons in the genome, right? They're huge outliers. And we just don't know about the sort of more subtle adaptations that may be happening because our current techniques aren't able to pick them up as well. And so I would say the work we're doing is more focused on trying to find these more subtle signals where it's multiple variants and multiple genes that are shifting subtly in frequency between populations that are driving the adaptation, as opposed to the one jackpot mutation.
I understand. Other questions? Yes.
If the disease inheritance can be any inference on fitness or any insight on fitness, the most frequent diseases are those that are not Mendelian in inheritance. So if you look at that in terms of bad fitness in the population as response to an environment, the majority of what we see as physicians are of complex inheritance. So that would be an insight, I believe.
So I'm so excited about what happened tonight. I really, really am. Oh, there's another question? Well, before that question, let me just say, Rasmus, fantastic, magnificent, great talk. And the others, I'm so proud to have moved here 6 months ago. So I'm really happy. Question, another.
So I work in animal models. And so when we work in animal models for developing therapies for human, we actually work with a really genetic defined background. So everything that we provide to medicine is actually really standardized. How actually basic science, and when we say animal model science, can help to explain some kind of variability. So should we then consider to repeat the same experiment in multiple divergent population and then give the results?
I would say the person who could speak to that best would be Michael, who has personally dealt with this issue in different strains of mice.
Yeah, I think you're headed in the right direction with that thought, because yes, humans are not inbred, as we know. These variants are in heterozygous form in many cases. And it's the combination of these different alleles in various forms, homozygous and heterozygous form across the genome, that form the genetic background that modifies the trait. And if you have a gene with a strong effect, a high penetrance or a strong effect size, that can be in different people or different strains of mice can have a very different expression, variable expressivity, we call that. And I have direct experience, which has been favorable and also kind of tragic, in that when you have a knock-in in a mouse model and you have it on the C57 black strain, which is the most commonly used in biomedical research, if you put that same variant on a different strain, you may have a very different phenotype. Now, is that a good thing or a bad thing? It makes it more difficult, more costly, more time-consuming to do the research, but we're dealing with a situation where now we're forced to look at variation, heterozygosity, different genetic modifiers, and since humans aren't inbred, that might be a plus. So I'm kind of going down the road of put in the extra work, put it at least on one other strain and see how that modifies your phenotype, and then try and look at the genetic variation that might modify that. Oh, that's a good question. I think the house mouse, the set of strains that house mice represent, come from the wild, and they come from more than one species or subspecies of Domesticus and Musculus, and perhaps do represent as a whole what you might find in the human population. So that would be a huge project to try and study that in a systematic way.
So part of that huge project's been done. There's been a large NIH effort to produce the collaborative cross mice and the diversity outbred mice. If you're familiar with mouse genetics, maybe you know these terms. But it's basically they took a huge number of traditional inbred lines used in research, and they've tried to sort of mix up their genomes, but in known identifiable ways. And it's a really fantastic resource for some of these translational things. I love mouse research. I think it's wonderful. And to go to your question, I think the absolute strength of working in these genetically identical inbred strains, knockout lines, is you really have the advantage of being able to test very clean hypotheses, provide very strong support. And then if you can connect that back to humans, you also have the ability to do really good translational. Because often what we've seen is that human evolutionary lessons tell us that, these pathways, these interactions, these networks are what's being perturbed, but they're probably not all druggable. They're probably not all things you would want to drug for any number of reasons. And so you can look at those networks and you can say, okay, well, what can we do? Understanding that this pathway is a problem, can we learn about it in a really systematic, controlled fashion and actually do something translational with this? Can we move it from a cool evolutionary story that we're not going to go and CRISPR everybody, and can we find something very actionable? And I love mouse work for that.
Well, again, thank you. It's been a wonderful, wonderful evening. This was a fascinating discussion. Great talk by Dr. Nielsen. We'll continue next month. I don't have the exact date. I know it's the end of April. April 24th. Herman Ponzer from Duke, and he's front page in January of Scientific American, the exercise paradox, why monkeys don't need exercise and humans do, and again, it's going to be a fascinating discussion. It's going to be a fun time. This was a great start. Thank you for starting us. Thank you. Thank y'all. As we say in the South, thank y'all. And thank you. Be safe going home.